Plants are mostly multicellular, prevalently photosynthetic eukaryotes of the realm Plantae. Directly or by implication, plants give nourishment, apparel, fuel, cover, and numerous different necessities of life. Mankind's reliance on yields, for example wheat and corn, is self-evident. Yet, without grass and grain, the domesticated animals that furnish individuals with nourishment and other creature items couldn't endure either. However, extracts of various herbs and spices from plants like thyme, clove, garlic, and others have been shown to have antimicrobial activity on several studies. In this documentary, we will explore and learn what are infections and bacteria, the antimicrobial activity of plants, and what is our research all about. We went to Quezon City Public Library in order to broaden our knowledge about infections and bacteria as well as the antimicrobial activity of plants. We have found 5 theses that are connected to what we are looking for, and 2 of these were the most relevant to our study, namely, effects of ginger on myofascial pain syndrome and antimicrobial effect of Andrographis paniculata nis or Sinta leaf extract against methicillin-resistant Staphylococcus aureus and Streptococcus pyogenes.
Sa unang thesis na nakita namin, we were able to gather data that using complementary and alternative medicine, which is CAM, has been growing and has attracted a lot of patients. With this, an old practice, which is the use of herbal treatments such as garlic, ginger, turmeric, and chili, have been achieved with renewed interest. The use of herbs as treatment has decreased the side effects of imposed and over-the-counter medication.
So, the second thesis na nagather namin sa library, which is entitled as Antimicrobial Effect of Andrographis paniculata nis or yung Sinta leaf extract against methicillin-resistant Staphylococcus aureus and Streptococcus pyogenes. Yung Andrographis paniculata is commonly known as Sinta in the local dialect, which has been primarily used for liver detoxification and minor alimentary tract infections. So, base doon, may mga observations din na yung powdered leaf is also being used against wound infections. Although majority of modalities in complementary and alternative healthcare have not been validated, it appears that examination of this new pattern is used and valid and justified. So, yung global growth ng CAM or complementary alternative medicine in recent years has brought a tremendous increase in the market share of food supplements and other natural products. Also, the pharmaceutical industry is venturing deeper on green drugs in order to synthesize the molecules and claim the intellectual property on the chemical form and action of natural substances. So, base din sa RRL ng thesis na yon, Shi and Wang on 2005 have also indicated that research showed that Xuan Xin Lian proved that A. paniculata decoction inhibited the growth of Streptococcus pneumoniae and alpha-pneumonic streptococci on laboratory tests, which is very relevant to our present study. Also, may nalaman din kami na yung flavonoids, which represent another group of bioactive compounds found in A. paniculata. So, ito yung mga phenolic compounds containing a broad spectrum of pharmacological activities like anti-allergic. These flavones have been reported to have antimicrobial, antibacterial, antifungal, and antiviral activities. So that is a very significant point nga, which shows that on a plant extract na may mga biocompounds na ganito, which is antimicrobial nga, which can be used to further deepen for future future studies. So for the preparation method of the A. paniculata leaves, yung grounded extract nun is ground using a Thomas Woolley mill with 116 grams and subjected to a cold percolation using 900 ml of methanol, sufficient enough to soak all the plant material. The percolation was covered and allowed to macerate at the room temperature for 24 hours. The percolator and material was rinsed with fresh portions of alcohol. Then after nun is sinalang siya sa isang agar plate para matignan kung ilang cm yung antimicrobial activity niya.
We were also able to find books which have a connection to what we are looking for, and the first is from Antibiotic Essentials, where it is said that infectious diseases are the leading cause of morbidity and mortality worldwide. The ability of bacteria, viruses, mycobacteria, fungi, and protozoa to cause infection is a balance between inoculum size, virulence, and the adequacy of host defenses. A medical doctor and the faculty member of Chinese General Hospital Colleges was able to answer our questions about bacteria and infections.
Okay, so bacteria is a part of a group of microorganisms, and we call them your microorganisms. So they are one-celled organisms. They have similar structure. Cell structure is similar to that of humans in terms of that we have both cell membranes and mitochondria and nucleus, etc. So they are considered as living. So these are bacteria. There are millions and millions and millions of bacteria in the world. Okay, some of them we haven't even discovered yet because they're still in nature. But bacteria is not all bad for our body. There are such things as your normal microflora. So these are the bacteria that live inside your body. And bacteria that live inside your body functions to protect you from other bacteria that can harm you. Some of these bacteria provide necessary nutrients as well. So you have a lot of bacteria in your body, and they're called your normal microflora. So there are a lot of normal microflora in your skin. There's a lot of microflora not just in your skin but also inside, in your internal. For the bad bacteria, it depends on, so if a bacteria is able to enter your body, so for example, it was able to breach the defenses of your body, one, it's able to breach the physical defenses of your body, your skin, or your mucosa, or your hair.
So, sa lahat ng information na nakalap namin, doon kami nakabuo ng topic which is entitled Antimicrobial Activity of Ethanolic Crude Extract of Selected Plants in Quezon City. The selected plants that we included were Psyllium guava or guava, Oreganum vulgare or oregano, and Laurus nobilis bay leaf. This will be extracted through the use of a solvent that will be tested for antimicrobial activity against bacterial isolate from skin.
Dr. Legaspi further emphasized the concept behind the antimicrobial activity.
Antimicrobial means these are substances that can kill or decrease the division or the cell division of your microorganisms. So there are different types of antimicrobial substances. We can call them drugs, okay? So we can also call them antibiotics. So there are substances or there are drugs that will kill the bacteria by inhibiting their cell wall. There are drugs that can kill the bacteria or kill microorganisms because it affects their DNA or RNA. There's some drugs that will kill your bacteria or microorganism because it affects their protein synthesis, or there are some drugs that kill the bacteria because of different, they affect the different enzymes that are in the bacteria. So these are the effects of your antimicrobial antimicrobials on your bacteria.
Guava's leaves or the bayabas leaves are known to have strong antimicrobial activity. So in this book, it was stated that these leaves has the antibacterial agent for wound healing. And the guava's bark and leaves are believed to treat or cleanse wounds. It showed that guava leaf extract has a good antimicrobial activity against Staphylococcus aureus or the skin bacteria. Guava leaves are proven that it can be a new source of an antimicrobial agents against Staphylococcus aureus.
Staph aureus is the normal, and Strep pyogenes are the normal microflora of your skin. Okay, so these are the bacteria that you usually find on your skin. So if, for example, I take a swab of your skin and put it on an agar plate, it will grow, and usually it will be Staph aureus or Strep pyogenes. Problem is, if your Staph aureus or your Strep pyogenes, if you have a skin cut and your Staph and Strep gets into the skin, then it is not in their normal environment, because their normal environment is the skin. It is not inside the skin. So when it gets to an environment that is not normal for them, then it will create damage. So your Staph aureus will now create skin diseases.
Therefore, the purpose of our study is to assess and compare the antimicrobial activity of ethanolic root extract of selected plants in Quezon City. And these selected plants are guava, bay leaf, and oregano. In order for us to develop this study, of course, we have to get leaves from the selected plants, which is the bayabas, oregano, and bay leaf. So these leaves must be dried, so para pag dried siya, mas madali siyang i-grind para maging powder. And then, kapag nakuha na natin yung powder na yon, we have to mix them with ethanol and water. Ethanol must be 95%, and then the water must be, for example, it's 2:1 ratio. So, yung ground leaves, we have to mix it with the solvent. Yung solvent nga, yung ethanol and water. And then, yung ground leaves natin, we must measure them by, for example, 25 grams nito, 50 grams, and then 75 grams para malaman natin kung alin yung may pinaka-effective para dun sa inhibition ng bacteria. And then next is, we must have agar plate para sa mga bacteria kasi ang agar plate ang nagsaserve as food ng mga bacteria. And then, i-grow natin yung bacteria doon sa agar plate. Saan manggagaling ang bacteria? Ang bacteria ay manggagaling sa skin at iyon nga yung tinatawag na Staphylococcus aureus. And then, kahit sa bare skin lang siya, pwede siya. Or sa may wound, pwede siya. And then, after 24 hours, mag-grow yung mga bacteria doon. And then, we have to use sterile swab. Sterile dapat. And then, i-aano natin dito sa skin natin. And then, we have to get the agar plate and put it in there. And then after that, after 24 hours, ilalagay lang din siya sa room temp na box. Dapat isolated siya. Tapos, doon, after 24 hours, makikita natin kung alin ang may pinaka-effective na extract. Makikita natin yung effectiveness niya doon sa inhibition. Ang meaning ng inhibition, kung gaano kalaki yung circle niya around the extract. And then, it will be measured by cm or millimeter. Since experimental study tayo, syempre, titingnan natin kung aling extract ang pinaka-effective. Anong gagawin natin pag nalaman natin kung alin na yung pinaka-effective? So, syempre, kailangan may magawa tayong product kasi nga capstone to. So, ang gagawin nating product ay soap. Kumbaga, since yun nga, galing sa skin bacteria, yung bacteria, pwede natin magamit yung soap as an alternative para sa mga bacteria.
you.